Hello, Assalamualaikum. My name is Nurusha Sabita Rizan and my metric number is S64301. My topic that I will present today is aquaculture biotechnology product, and the topic that I choose from Oceania is transgenesis or transgenic. This is the content that I will present. First is introduction, method, advantages, disadvantages, future prospects, and conclusion. Introduction. Transgenesis or transgenics may be defined as the introduction of oxygenous gene or DNA into host genome, resulting in its stable maintenance, transmission, and expression. The technology offers an excellent opportunity for modifying or improving the genetic traits of commercially important fishes, mollusks, and crustaceans for aquaculture. Okay, in this transgenesis, we have four method. First is micro injection, second is electroporation, third is per-mediated gene transfer, and the fourth is retroviral infection. Now we go to the injection. Microinjection method has been used successfully in the production of transgenic fish, and it is a commonly used technique due to its simplicity and reliability. The use of microinjection method results in higher survival rates for manipulated fish embryos than the electroporation method. The most established method for gene transfer in fish is microinjection. Microinjection that allows delivery of the transgene directly into the nucleus. Transgene is directly microinjected into the male pronuclei of fertilized eggs. Transgenic technology through DNA microinjection into fish embryo has made great gain in the last decade. It is shown that the DNA injected into the cytoplasm of fertilized fish eggs could integrate into the fish genome and be inherited into the germ line. Now we go to the electroporation. Electroporation method has been shown to be the most effective means of gene transfer in fish, since this method can treat many fertilized eggs in a short time. Electroporation utilizes a series of short electric pulses to permeate the cell membrane that make possible the formation of temporary pores on the surface of the target cells, through which the transgene is introduced into the cytoplasm, where it is then delivered to the nucleus by the cellular machinery. Electroporation has been preferred in many laboratories for gene transfer in fish system because of its efficiency, speed, and simplicity. And we go to the embryonic stem cell. This is a powerful strategy for transgenesis. Cells are removed from developing blastocysts and are grown in culture in an undifferentiated state. Foreign DNA is introduced by electroporation, transfection, or microinjection into the stem cells, and the cells then reintroduced into the blastocyst, which is then allowed to develop to term. If some transgene containing cells develop into germ cells, subsequent breeding will produce transgenic individuals. This method allows for the pre-selection of cells and increasing the concentration of cells containing the integrated oxygenous DNA before introducing them into the blastocyst, thus increasing the chances of producing transgenic individuals. For example, selection can be conducted by fusing the gene of interest with a new gene, which then allows the cells that incorporate this recombinant gene to grow in media containing G4I8, which is normally toxic to cells not possessing the gene. And the last method is retroviral infection. Retrovirus efficiently integrate their genetic materials into the genome of the infected cells by a precisely defined mechanism. Only a single copy of the provirus is inserted at a given chromosomal site, and rearrangements of the host genome are not induced, unlike other methods such as microinjection, where foreign gene insertion may be, may be in multiple copies or rearranged as in sperm mediated transfer. The foreign gene is incorporated into the viral genome, which is then transferred to the host by viral infection. To date, no fish retrovirus has been isolated. The retrovirus is often species specific, which are available and well characterized, have been assumed to be ineffective on fish. However, research show that a pseudotype pentropic retroviral vector originally developed for human gene therapy was able to infect zebrafish cells and embryos, respectively. The latter microinjected a retroviral vector pseudotype with the enveloped glycoprotein of the vascular stomatitis virus into the blastula stage, and 8 out of 51 founders transmitted troviral DNA to the progeny. Now we go to the advantages. Transgenic fish have been developed for application such as the production of human therapeutics, experimental models for biological research, environmental monitoring, ornamental fish, and aquaculture production. Growth enhanced transgenic fish have improved feed conversion efficiency, resulting in economic and potential environmental benefits such as reduced feed waste and influence from fish farm. Transgenic strains provide useful model system for studying the consequences of growth enhancements from genetic, physio- physiological, and ecological standpoints. The trait of growth rate has received the greatest attention because of its importance in aquatic animals production system. Commercial production of transgenic fish able to transmit desirable characteristics such as enhanced growth, or disease resistance to their progeny. Growth acceleration in fish has been one of the targets of gene transfer experiments. Uh, th- uh, Second, the faster growth of each generation can lead to increase. I'm sorry. The faster growth of each generation can lead to increased production per unit time, along with savings on total food profile of meat produced. Use of transgenic fish as bioreactors for the large-scale production of rare human therapeutic proteins or novel foods for specific dietary requirements. Transgenic lines of tilapia engineered to produce human clotting factor 7, which is used in liver transplant and in treating injuries. Zebrafish, genetically modified to manifest a defective aortic valve development, has made it possible to identify the role of an enzyme, UDP glucose dehydrogenase, in the process of the embryonic development of this valve. Transgenesis is proposed as a mean for removing allergenic substance in seafood. Fish are recognized as test organisms with distinct and superior benefits in providing insights to disease processes. And the last one is transgenic fish have been used to study developmental regulation of genes, improve cold tolerance, increase growth rates, and improve feed utilization. For the disadvantages, the primary ecological concern regarding utilization of transgenic fish are the loss of genetic diversity and loss of biodiversity and reduction in in species richness. Number two is concerns range from interbreeding with native fish population to ecosystem and effects resulting from heterogeneous competition for food and prey species. And the third is only about 1 out of every 100 eggs micro injected will stably incorporate the recombinant DNA sequence into its genome and subsequently transmit the transgene to its progeny. For future prospect, future prospect, undoubtedly, transgenic fish technology has a great potential in revolutionizing the aquaculture industry by introducing desirable genetic traits into finfish, shellfish, or crustacean. Superior transgenic strains with improved genetic traits can be produced for aquaculture. This trait conversion efficiency, resistance to some known disease, tolerance to low oxygen concentration, and tolerance to sub-zero temperatures. And for the conclusion, there are many applications of transgenic fish technology in aquaculture and biomedical research, and several major problems that need to be solved in order to utilize this technology efficiently and safely. Further research should work with the DNA sequences of fish rather than mammalian origin to increase public acceptance and avoid sequences of bacterial or viral origin. Transgenic technology is developing rapidly. However, consumers and envi- en- environment, envi- environmentalists remain cautious of its safety for use. Research is needed to ensure the safe use of transgenic technology and thus increase public assurance. And this is the reference that I use. Thanks.